Okay, and welcome to the fourth lecture on the MCAT Behavioral Science Review. In this lecture, we're going to be covering two chapters, Chapter 10 and Chapter 11 from your manual. Chapter 10 is called Behavioral Neuroscience, and Chapter 11 is entitled Sensation, Perception, and Cognition. Chapter 10 is actually very short, and it covers some very general things regarding neuroscience and the tools used by the field of neuroscience itself. And then we'll spend most of our time afterward covering the material on sensation, perception, and cognition, which will overlap quite a bit with the material you have had in anatomy and physiology. To get started with in Chapter 10, what we're going to do is just take a brief look at some of the early techniques by which the brain has been studied. So you really do not get any significant amount of scientific work in the field of brain science until at least the 1800s. And the reason the 1800s is the point where it starts is largely because medical doctors primarily, well before the point that psychology begins in 1879, start evaluating individuals' patterns of behavior and correlate the types of disorders that individuals have with the tissue damage that they can verify through anatomical work after the person dies. So throughout the 1800s, what you wind up seeing is medical doctors paying families to do autopsies on individuals that have had strokes or other types of seizures, in a most general sense. So you have an individual, he's fully functioning, there's no general problem, and then one day he has this quote-unquote seizure event and can't speak any longer. Certainly that person will be brought to the doctor. The doctor recognizes that there's probably something wrong in the individual's brain, but of course you can't do anything about that because it's the 1800s. So you pay the family a fee so that you can do anatomical work on that individual after he just dies from natural causes. And when that occurs, the medical community starts correlating tissue damage in certain areas of the brain with the types of deficits that they saw existing while the person was alive. And that's actually kind of an extension of brain mapping work that was done as early as 1825. And no, that's not a mistake. 1825, brain mapping work on dogs was being done with simplified electrodes, that dogs would be anesthetized, the top of their skull would be removed, and the surface of their cortex would be stimulated in a systematic fashion to see if the anesthetized dog would actually exhibit any patterns of behavior like leg twitches and so forth. And of course, obviously, they would find that out, but you could see that kind of work, which was, I believe, primarily done in France, kind of led to the idea that if a person has some sort of seizure event during life and then they can't use their left arm any longer, that there's actually damage to a specific portion of the brain, and that's why this whole process of paying for the autopsy takes place, because as you might imagine, even today, a lot of people don't want to donate their bodies for anatomical work after they're dead, especially for just straight scientific work. But some of the instances in where this happened were instances of language deficits that were developed, of course, by Broca and Wernicke, recognizing Broca's and Wernicke's area, which are related to language disorders. Phineas Gage in 1848 stands out as a primary one in the field of psychology because Gage, as you probably are familiar with, was using a tamping iron for explosives while he worked on a railroad in Vermont, and made a mistake during a fist fight that broke out on his work crew. he kind of got a little confused and didn't take proper safety precautions and was tamping down the gunpowder, and it exploded and blew the metal shaft right through his skull. As you see the image here on the screen, blew through his left cheek and out the top of his skull. And that's a picture of Phineas Gage over there actually holding the tamping iron sometime, I believe within a year of the time that it occurred, that photograph was taken. But the issue with Gage was that he lost his ability to control his emotions, and his emotions would swing wildly throughout the day. People could not stand to be around him any longer as a result of the manner in which he would behave. And this was all very different from the very reserved individual who had made it to a supervisor on this train crew. So it was a big difference, and certainly at the time it wasn't clear what had happened, but that story remained for decades until, in the 20th century, you could really start to kind of put together what may have actually happened. So strokes, closed head injuries, they all, kind of fit into the same way, at least for the 1800s, where individuals would get closed head injuries, seizures, or other types of things. And, when I say seizures in the most general sense, back in the way it would have been used in the 1800s, that there was an event that occurred. That's more or less what was being said. And then to do anatomical work on those individuals after they died to find out the location of it, keep track of this information in the medical community, and then to build up a picture of which sections of the brain were actually controlling which aspects of behavior. Now, of course, now we have neuroimaging techniques, and a wider and wider range of them all the time. And one of the first ones to develop was based off the X-ray, computerized tomography, or the CAT scan, or CT scan. The CAT scan is nothing more really than a three-dimensional X-ray. And as you might imagine, although it has been used for the brain to evaluate where damage may occur, what you're really looking for in terms of those X-rays are areas of the brain that show up differently because the sections of the brain have actually died off as a result of some sort of injury or internal bleeding in the brain and so forth. Computerized tomography is, I believe, mostly used for non-neural tissue things. Although it is certainly still used, there are better techniques at the current time than a CAT scan for your brain. And that's largely because the newer techniques allow you to evaluate the brain while it's functioning, whereas a CAT scan is a static image, right? It's so if somebody were, for example, shot in the head with a shotgun and there was a bunch of pellets, buckshot, residing throughout the brain, a CAT scan would make a lot of sense because you could identify where the buckshot was, and then the surgeon would know in which direction to try to remove each of the individual pellets. But if you have an individual who has had a stroke today, finding where the damage is only going to take you so far when you know that you can use a separate method which would allow you to try to get the person to activate that area of the brain and the other areas around it in a functional process. And so oftentimes the term functional shows up in the names of these neuroimaging techniques. Functional always refers to a measure over time, where essentially you could take a video of the data. Unlike a CAT scan, which is like a snapshot, a functional process, you can take a video, and so you can evaluate the activity or the function of the brain over time. Magnetic resonance imaging is also a static process. And in fact, most of these neuroimaging techniques are actually based on the magnetic properties of blood in your brain. So blood has magnetic properties, and when the blood passes through your brain, because that's the area that you're trying to evaluate, what you're typically doing is evaluating for the amount of magnetism from certain areas of the brain based on the large magnet that you place the person in the machine. So magnetic resonance imaging evaluates the areas of the brain that are different in terms of the amount of magnetism. And a greater magnetism indicates a greater blood flow to that particular area. The MRI, though, is also a static image. But there was an advance with the MRI to the fMRI. If you hop down a few lines there, the functional magnetic resonance imaging, which is the same thing as the MRI, except now it's essentially video format, that you could read it across time. So you still are following the magnetic properties of the brain, except now you can have a person perform different types of intellectual tasks to get the blood flow in separate areas of the brain. Because it has been a basic assumption that the area of the brain that is active requires more energy, and therefore there's going to be greater blood flow to those particular areas of the brain to deliver more glucose as an energy source. So the vascular system, the arteries and the veins, they will constrict and dilate to manage the amount of blood flow to particular areas of the brain. And for the areas that need it, they will dilate, bring in more blood, and therefore more glucose will be in those areas. And what you're really doing with the fMRI, though, is still measuring the changes in magnetic properties of areas of the brain based on the change in blood flow because a change in intellectual task takes place. The earliest of all of these measures was the EEG, the electroencephalogram. It was not until sometime around 1930, I believe, at Harvard University, where the first usable EEG was created. In fact, as I recall from looking up the history on EEG at one time, I believe it was theorized as a tool possibly in the late 1890s, approximately 1900. And it took about 30 years roughly to actually build a usable EEG. And the very first one, the only one that existed at the time, was at Harvard University at around 1930. So as you might imagine, the EEG was primarily used for medical purposes for people with brain damage. Not a lot of work on the field of psychology was actually done at the time because as we've even seen in the previous lectures, you don't really get much in the way of behavioral biology or physiological psychology, which were kind of the terms used at the time, until around the 1950s. So back in the 30s, the EEG was primarily used for individuals that clearly had brain damage, seizure activity, or other types of neurological problems that the medical community could kind of work on. The EEG is itself a rather crude measure. It's easy to use. It's one of the cheaper ones that you can gain access to, but it really does not do a great job in terms of distinguishing electrical activity, largely because the pads that get placed on your skull are over top your the scalp, your bone, the cerebrospinal fluid, and then are detecting the electrical activity of all the brain area underneath of it, right? Actually, and not all the way to the center core of your brain, of course, but it's not like the EEG is reading the electrical activity from a single neuron or just a couple of them that are located together. What it's doing, given that neural cells are so small, a single pad in an EEG is simply reading a gross measure of electrical activity in a general area from where it was placed on the skull. And that, of course, as I've mentioned, is being degraded as a result of the amount of material it has to go through to be able to read the electrical activity, on top of the fact that the size of the pad and its ability to be sensitive to changes in electrical activity are going to largely affect how it reads things. What you're really doing with the EEG is just getting a very general measure of what's actually happening in one general vicinity. It's almost like if you came over to New York City in a spaceship, and the spaceship is completely quiet, and what you did is you took a microphone and you let it slide down a wire so that it was over top of Queens. And then you measured the amount of noise coming from Queens. And then you took your spaceship and you flew it over to Manhattan. And then you hung the microphone down and you measured the amount of noise in Manhattan. And, you do this to all 5 boroughs in New York City. And then what you wind up concluding is that the center of activity of New York is Manhattan because it's the noisiest area. And if you said this to somebody, people would look at you like you were stupid and say, well, why are you using such a crappy measure? Well, that's what the EEG does. And, it did what it could do at the time that it was built. There's better techniques now, but it's, of course, low cost. And one of the things that you can actually get out of the EEG is if there's global changes in functioning, which it turns out there actually are. So there are literally waves of electrical activity that travel across the surface of your brain. So a wave, that is being essentially a line of electrical activity that travels over the course of your cortex. And if you have these electrical pads from the EEG on the surface of your scalp, you can measure that kind of global activity. And the EEG's recognizing its presence, and it's doing it fairly cheaply. So it's valuable in that sense. But at the current time, you really have to start getting down to smaller units, smaller sections of brain, even if not individual neurons, to be able to understand what's actually going on, because most of the global activity has actually already been looked at, and there's only so much you can actually do with it theoretically, and in terms of your ability to help somebody with a neurological disorder. So you have to keep drilling down into smaller and smaller units. So the EEG has largely served its purpose. It will probably disappear and be replaced by better techniques sometime in the coming decades. The magnetoencephalogram, as you might imagine, is also based on the magnetic properties of the blood in your brain. But in this particular case, you are actually measuring the blood flow in certain areas in a way that is a bit more specific than you saw with the fMRI, largely because the fMRI does not have as good temporal resolution as the magnetoencephalogram. Okay, so for example, a fMRI might take one image every 0.2 of a second. And magnetoencephalogram is actually going to give you a better, more continuous reading of changes in electrical activity based on the magnetic properties of the blood itself. So the magnetoencephalogram largely is probably going to replace the fMRI. But the fMRI, as an older technology, is of course less expensive. And so it'll be around until you get that basic shift in cost versus value, which plays itself out in terms of just the capitalism of the whole process. And positron emission tomography, that one is, I believe, about 40 years old. Positron emission tomography requires an individual to be injected with radioactive glucose. So once again, right, you're back to measuring changes now with the blood. And in the earlier measures, like the magnetic resonance imaging, you were looking for changes in magnetic properties based upon the pooling of blood in certain areas of the brain. You are relying on that change in volume of blood in certain areas of the brain by injecting the person with radioactive glucose, so there is a greater statistical chance that the radioactivity will come from an area where there's a greater concentration of blood, and therefore that would be the area identified as more active at that current time. Of course, all of these measures are internally consistent with each other. They all reflect the same kinds of changes or activity in the brain overall. Most people don't like the idea of being injected with radioactivity, despite the fact that it's a very low-grade alpha radiation and is not going to be harmful. But I imagine, again, the MEG will probably replace the PET overall. But there may be other more specific medical differences between them that I'm not fully aware of. But as all things, there are certainly changes in this technology all the time. Much more work being done on biophysics, and specifically individuals working on creating new types of scanning devices for medical use. And there's just going to be a continual change and development of new techniques. That gets us into a very general discussion here about the anatomical changes in the nervous system, or anatomical organization of the nervous system. What you're largely looking at here is the comparison between the sympathetic and parasympathetic nervous systems. This is something we're going to wind up looking at again later on in the next chapter, which is part of this fourth lecture. But overall, this is a straightforward discussion of the difference between sympathetic and parasympathetic nervous systems that comes right out of anatomy and physiology. And the basic story is this. Your sympathetic nervous system controls arousal, and your parasympathetic nervous system controls relaxation. If you can't keep that straight, the simple way to remember it is, for a person that's excited or upset, you would have sympathy for them, and therefore that's the sympathetic nervous system operating. So when you step into the street and you almost get hit by a car and you jump back and you're breathing heavy and you're thinking, oh my God, that's your sympathetic nervous system. That's when you would have sympathy for somebody who had experienced something like that. Now, the sympathetic and parasympathetic nervous systems control the exact same internal organs, but they do it in opposite ways. The sympathetic nervous system is largely going to allow for blood flow to move away from internal organs and out to your limbs so that you can control your muscles in a fight or flight scenario, like getting back on the sidewalk so you don't get run over by a car. And when you're relaxed, more blood flow will go into the internal organs so that you can digest food and perform other types of internal medical processes that are necessary for your normal health to occur. One thing that you have to keep in mind, and I believe we were going to hit this point again in the next chapter, but just to get through it now, although the sympathetic and parasympathetic nervous systems are connected to the same internal organs, the sympathetic essentially is connected to the center 2/3 of your spinal cord and then gets wired out to those internal organs, whereas the parasympathetic, para always meaning side, is wired to the top and the bottom of your spinal cord and then gets wired to the same internal organs. And that's why the name sympathetic, or at least the name parasympathetic, is used in this case, because the parasympathetic nervous system is literally on the sides, the top and the bottom of the spinal cord, whereas the sympathetic nervous system occupies the center 2/3 of the spinal cord. Now, some of the general anatomical organization of the outer surface of the brain, as what we're going to talk about now, you do see further discussion of this in the next chapter as we cover brain activity. But in terms of psychology, and almost certainly questions that you're going to wind up seeing on the MCAT, you're going to deal with these primary sections of the brain. For absolutely certain, the outer surface, the lobes of the brain, and the general makeup of the sections of the outer surface of the brain are clearly going to be questions, as is the construction of the primary motor cortex and the primary somatosensory cortex, as you see in the bottom center of the slide itself, and to a lesser degree of relevance to psychology, the brainstem. Because as you probably know at this point, as you go along the brain toward the spinal cord, you simply get toward older and older sections of the brain that primarily deal with just life-sustaining activity. And so in terms of the brain stem straight up to the thalamus, there's nothing really there to any great degree that is specifically related to human thinking, culture, language, or any of the higher order functions of human endeavors. All that stuff in the brainstem basically keeps you alive and just manages some low-level integration of sensory information. It's not like it's not important, of course, but it is not related to the higher order of thinking, which is usually the kind of stuff that the field of psychology will wind up focusing on. You know, language and culture and human interaction and so forth. Most of that stuff resides on the outer surface of the brain itself. So you should be aware when you take a look at the outer surface of the brain that there are 4 general lobes, and those lobes, when you take them clockwise, there is the frontal lobe, the somatosensory lobe. In the back, there's the occipital lobe, and then the temporal lobe. Those are the 4 main lobes. The cerebellum is not a lobe. It's a section of the brain that, in a general sense, primarily deals with motor function and motor coordination. So it's not considered a lobe, but it's relevant primarily for motor activity. It does other things, and as you'll see, certainly with the brain, there's a lot of integration in the brain, so no one area only does one thing. That's absolutely something that you should be very aware of. You know, for an undergraduate in a general psychology class, you know, I'll tell them that the frontal lobe is related to behavior, and I won't go much into it in detail, but the frontal lobe is integrated with all the other lobes, and you know, if you get damage in the frontal lobe, you could wind up affecting vision or hearing or touch or smell, things that actually exist in other lobes primarily because the brain is integrated and signals travel all over the darn place. So causing damage in any one location almost certainly will make it seem like some of the damage exists elsewhere, and you have to be very careful about that. So it's kind of a thing to be aware of most of the time whenever there's a discussion of the brain. a simple description of which section of the brain does is usually primarily identified. Now, in the back portion of the frontal lobe, there is the somatosensory cortex, which you actually see in the bottom center of the screen itself. The somatosensory cortex exists kind of as a semicircular ring across the top of your head, and there is a specific anatomical layout that describes which sections of your brain are controlling which sections of your body. So as you might imagine, as electrical signals travel around your brain and get sent out to control your muscles, you can always go to the primary somatosensory cortex, which is the very last place any kind of circuitry where we'll say decision making, and I got to air quote that, decision making gets done with respect to a pattern of behavior that's going to exhibit. And if that decision making always takes place in the primary somatosensory cortex on the top of your head, and then let's say sends a signal down to your foot to wiggle your toes, you can, in the laboratory, always measure the electrical activity associated with your feet so that you see that it occurs always about 1/10 of a second roughly before your toes actually move. But this is back in the 70s when this kind of technique was developed. Everybody got all excited and they were saying, oh, look, I can tell what a person's going to do before they do it. But okay, and that's roughly true, but you have to recognize that your nervous system is nothing more than a circuitry. And all you're doing is going backwards in the circuitry to find some of the signals that show up on particular pathways that are going to go to your foot, right? The opposite side of your body. So as the signals travel through the area of your brain that controls your foot, you can detect it just before you actually see the foot move, and the time in between those is the length of time it takes the electrical signals to travel through your nervous system, from your brain through your spinal cord and then out your peripheral nerves down to your foot. So, in the end, you're just looking at different portions of the circuitry. When people start talking about, this is a mind reading technique, or, we always know what a person's doing before they actually know it. Yeah, kind of technically, sure you do, but of no great importance. If you were to get in there to try to redirect a person's thoughts so that the electrical activity in the brain was making the decision for the person to do something, but they weren't consciously aware of it yet, you would have to have some sort of fantastic supercomputer intervening on that section of the brain to modify which electrical signals were being generated so they actually did something else. And that's just technology well beyond our capacity at this point, right? To have some supercomputer that fast with the programming in it to detect patterns of electrical activity, to know that the person's going to say the wrong thing, but instead the supercomputer analyzes that and then injects electrical signals through electrodes into other pathways that overrides the inappropriate response, and then the person says something nice. That's all science fiction at the current time. It's a kind of a straightforward extension of what we're talking about, but it is itself beyond our capacity now. Neurotransmitters are, of course, important. One of the things about neurotransmitters that I always point out to my students is that having different neurotransmitters throughout the brain largely electrically isolates particular pathways that are right next to each other. And that largely is because neural cells have a basic rule, and that rule is cells of one type of neurotransmitter almost only communicate with other cells that have the same neurotransmitter. Okay, it's not a 100% rule. So a cell that has dopamine will communicate with other cells that have serotonin or GABA or acetylcholine, for example. But most of the time, and I say this generically, this is not a real number, greater than 90% of the time, any dopamine cell is only going to communicate with another dopamine cell. And as a result, that electrically isolates the signal or the pathway through the brain because the cells won't communicate with other cells that are literally right next to it that are operating with another neurotransmitter. So that seems to be primarily the issue. Now, of course, it's been learned throughout the 20th century specifically that you can modify the activity of the neurotransmitters in particular ways by ultimately increasing or decreasing their activity, or even injecting an entirely new substance that will function similar to the pre-existing neurotransmitter. So for every drug that has some influence on the neurotransmitter, on the neurotransmitter system, it clearly must have a shape similar to the originally occurring neurotransmitter that exists there, because the receptor for that neurotransmitter is the only way to get the cell to receive that new substance. And if you throw in I'm going to do this in a hypothetical, ridiculous sense. If you created a literal box, some sort of molecule that was literally shaped like a box, it was just a square, tiny, tiny squares, and you injected those into the person's brain, into the area that had dopamine. A cube, right? A molecule shaped like a cube is completely differently shaped than a molecule of dopamine. So those cubes would be in the synaptic junction, but they would have no bearing on the receptors. The receptors couldn't deal with a cube molecule. It's not designed to receive a cube molecule. So those cubes would just float around and do absolutely nothing. To get a drug to do something in the brain, it's going to have a shape that is similar to that pre-existing neurotransmitter, or if you want to say, a shape that will fit onto the receptor itself, which is, of course, based on that neurotransmitter. So all those things are related to each other. Now, there are literally over 100 different neurotransmitters found throughout your nervous system, but there's only approximately 7 that typically have an influence on psychological properties. And those are listed here. And so what you generally see is some overlap between the activity of the different neurotransmitters. So it's not quite like you can spend your career studying dopamine to get people to wave their hand. You know, the neurotransmitter is not so specific to a pattern of behavior. It's usually related to emotional functioning, or in a very general sense, a broad basis for increasing or decreasing a general pattern of behavior, but it's never going to be very specific. In fact, the manipulation of neurotransmitters overall is a fairly gross type of influence, not very specific to particular patterns of behavior, as you'll see here. Like, for example, dopamine is used for reward and positive mood. And reward and positive mood go together. They're correlated on their everyday function. However, reward and positive mood are literally distinct circuits. And so if you wanted to, in a treatment for someone with a psychological disorder, increase positive mood but not reward them, have the increase of the drug act as a reinforcer, or if you want to do the reverse, you're kind of stuck. Dopamine is going to wind up affecting both of those systems as well as other systems as well. This table that you see on the screen is just giving you a general description of what these neurotransmitters typically do, but it's not like it's their only function in all places in the brain, or in all places of the body. So, this table gives you a general description of what a neurotransmitter does in the same way that I say the frontal lobe is related to motor behavior. And yes, it is, but the frontal lobe actually does other things as well. So, in a brain that is so complicated and so integrated, we just speak about it in very general terms. Anytime you do actual research on the brain, you have to highlight specifically particular areas or pathways to know exactly what you're talking about. But you can't talk about, the role of the frontal lobe in terms of motor behavior any longer. We're well past that point. That would have sufficed back in the 1940s or 50s, but not today. So if you take a look at the variety of neurotransmitters in this case and the general things that they are related to, that will probably give you some general idea regarding kinds of questions you would likely see on the MCAT. I would expect there to be questions regarding which of these neurotransmitters are related to increasing a person's mood if they had depression. And you should know that it would be dopamine. And if it was a question about motor behavior, almost certainly it's going to be related to acetylcholine, because all of the outgoing messages from the brain that travel off the neuromuscular junctions are all, 100% of them, controlled by acetylcholine as its neurotransmitter. And so any question about motor activity, almost certainly the answer is going to be acetylcholine. Anything related to positive mood, almost certainly is going to be related to dopamine, but depending upon how it's written, you might see norepinephrine or serotonin as a possibility. If you stick with this table here, you're going to get more distinctions, like serotonin being more related to memory rather than dopamine does, although serotonin is related to relaxed mood and dopamine positive mood, and usually relaxed moods and positive moods are co-varying, so it would be hard to distinguish between those two. So if you had a question about mood and neurotransmitters, you'd probably have dopamine as an answer or serotonin as an answer, but I think it would be wrong to put dopamine and serotonin to ask a question about positive or good mood, because that just makes confusion between the role of the neurotransmitters in that case. Otherwise, just look through these and in general get an idea of which ones they're related to, and we'll move on from there. The final slide in this particular chapter is related to some general areas of the brain. For example, anytime you hear about the limbic system, the one and only thing you should ever think of is emotions. And in fact, that's a reasonably good one to actually connect together. The limbic system is a system that looks like two ram's horns that kind of come off the thalamus in the very center of your brain and then wrap around in two big curly cues, one on the left side of your brain, one on the right side of your brain. And that curled section of brain contains all of the possible emotions that you can exhibit. There are no emotions outside of the limbic system. The primary areas that control emotion are the limbic system themselves. And if you travel along the limbic system, a variety of emotions exist next to each other along these curled pathways. So it is possible, from the standpoint of biology, to activate two emotions that may seem completely different, like happiness and sadness at the same time, because happiness and sadness are not operated by a single section of emotion of, let's say, happiness. And when it gets really active, when the happiness area gets really active, you get happy, and then when it becomes less active, you can become sad. It does not work that way. Happiness is a section, Sadness is its own section. You could turn them both on at the same time, or you could turn them both off at the same time. The fact that human beings always recognize in themselves mixed emotions, like they'll say things like, gee, I really love my grandmother and I'm sorry she died, but I'm happy she's not in pain anymore. And so in that case, what they're recognizing is they're both happy and sad at the same time at their grandmother's death. But for different reasons. Happy she doesn't feel pain anymore, sad that she's passed away. And that's why you can feel multiple emotions like this, because all of your emotions are distinct sections of the limbic system itself. At the very end of that curled section is an area called the hippocampus. So if you pretend that the limbic system is shaped like two ram's horns, one on the left and one on the right side of your brain, and you go up to the ram and you take a large cork and you pop it onto the end of the horn so that the ram doesn't stab you, that cork that covers the tip of the horn, that's the hippocampus. That's just the way it's built. It's right at the very end of the limbic system. And the hippocampus is primarily related to memory function. So as you might imagine, if the hippocampus is connected to the string of all the emotions you can experience, then the hippocampus is going to be modified by emotion very much. And to say that another way, which makes it sound very obvious, emotions influence how well you remember things. And that is a statement that I think you should recognize immediately is obvious. And it's true because of the underlying biological structure of your emotional system and memory system, because they are literally connected together right next to each other. Any two areas of the brain that are closer together are always going to find it easier to sprout out axonic or dendritic tendons and to be able to connect to that other area. Areas in the brain that are further away from each other are always going to have a harder time connecting each other, and so you're going to be less likely to see that. So because the hippocampus is connected directly to the limbic system, it's not a surprise that emotions modify memory, and I would not be surprised if that wasn't a question. And now the prefrontal cortex is related to executive function. Now, we kind of have to define this a couple ways. Prefrontal cortex is part of your frontal cortex. It's part of your frontal lobe, right? So the frontal lobe is the front lobe on the front of your brain. Now, the prefrontal section is just the back portion. That's all that is. And the prefrontal cortex tends to be along the sides. So if you have to move back toward the top of your brain along the sides, that's generally what's referred to as prefrontal. The frontal lobe, as I mentioned, primarily is related to motor behavior. And for you to make decisions about how you're going to behave must involve a lot of the frontal lobe, because your decisions about what you're going to do next are always decisions about overt behavior. And overt behavior is being managed by your frontal lobe. So executive function, which is a term from the field of memory that refers largely to your highest decision-making process. This would be most closely associated with the little voice in your head when you concentrate specifically and think through a problem, like you're doing a calculus problem, or you have a complicated social problem that you're trying to solve, like how do you get your ex-wife to come to your new wedding with your children there, and people are going to dislike each other, and there's going to be a lot of snarky activity. How do you manage this complicated scenario? And you sit down and you think it through, and it's really difficult. That thinking, that little voice in your head, is formally referred to as executive function. It's a term that's come out of the field of memory largely because it's an extension of your short-term memory and a variety of other memory function systems. So it's the term that's been developed, so you can just accept it. But it's essentially the voice inside your head. Now, because that voice in your head, the executive function, is primarily related to determining which actions you're going to perform next, it's no big surprise that it's connected to the prefrontal cortex, which is just a portion of your frontal cortex, which is, of course, related to the determination of which pattern of behavior you're going to exhibit next. So no big surprises here. It's kind of a lengthy series of connections in this case, but it's all very straightforward. And the last term in this case, before we finish up this particular chapter, is neuroplasticity. Neuroplasticity is, of course, your neural cells' ability to rewire themselves. And why do your neural cells want to rewire themselves in your brain? Well, because your brain is where all your experiences leave their trace. And the traces that they leave in your brain are literally the pathways of information that travel through your brain. So during the course of your day, just like today, any other day, you experience things, you have good events, bad events, boredom, happiness, you exercise, you lay on the couch, you have all sorts of experiences. And literally every single thing you do affects the wiring of your brain. And so as your brain changes over the course of the day, and literally as well over the course of the evening while you're dreaming and your brain is rewiring itself, you wind up saying that the manner in which your brain rewires itself is called neuroplasticity. It's not so much typically that your brain is generating or decreasing the number of brain cells it has, but rather the tentacles of the dendrites and the axons, right? The number of potential dendritic pathways and axonic pathways, which can increase and decrease. The number of dendrites or number of axonic branches can increase or decrease, thereby always changing the total flow of electrical activity through your brain, and that's what ultimately changes your behavior come the following day. So cell growth and repair is going to be extremely important for any kind of neurological disorder that an individual has. For Parkinson's or Alzheimer's disease, where you have cell damage occurred naturally as a result of genetic processes, you want to get in there and regrow those cells, and then all of the symptoms of those diseases will go away. If somebody gets clubbed in the head with a baseball bat and then they have a large section of their cortex which dies off because of the damage, you want to be able to go in there and regrow that area. Now, central nervous system tissue naturally does not repair itself very well. Maybe, and this is again a ballpark number, 5 to 10% of central nervous system tissue damage will somewhat repair itself, but most of it does not. And that's where you're looking at stem cell therapy. Because if a person has a broad range of damaged area in their brain, for whatever reason the damage exists, you want to get in there and grow new cells. The only way that we know to grow new cells is by activating some of the basic neuroplasticity processes that already exist in the cells themselves that are generally just not naturally being activated. Some of that comes from stem cell therapy to throw in new cells and let them grow and subdivide and start taking over the area of the damaged section. In other instances, there's a variety of ways to try to stimulate the underlying neuroplasticity from the cells themselves so that they grow new branches and possibly take over the section of brain that is permanently damaged as a result of some sort of disorder. And that largely covers the basis of what we're covering in Chapter 10.